Everybody, to our third installment of our GCE for All International Webinar Series. Today we have two excellent scientists, Dr. Edwin Antony and Dr. Chris Ahern. First up today is Dr. Edwin Antony, professor in the Department of Biochemistry and Molecular Biology at the St. Louis University School of Medicine. Dr. Antony earned his Bachelor's of Science in Zoology and Chemistry at Loyola College in India, and then a Master's in Biochemistry at St. Joseph's College, also in India. He then came to the States, where he earned his PhD in Biochemistry and Molecular Biology at Wesleyan University in Connecticut. He started his professorship at Utah State University in 2012, then moved to Marquette University in 2015, where he was promoted to associate professor in 2019 before moving to St. Louis University, where he currently resides. Dr. Antony has been very successful in obtaining funding for research programs, including 2 NIH R01s currently, and having successfully completed several major NSF and NIH awards previously. His lab focuses on the molecular mechanisms underlying DNA repair and recombination, specifically how enzymes recognize damage in DNA and coordinate with the cell cycle and replication machinery to coordinate these damages. Other projects include studying mechanisms of electron transfer and oxidoreductases and how large conformational changes in these complexes coordinate these reactions. And so we're very excited to have you here today, Dr. Antony, and to hear more about your research. So the floor is yours.
Oh, thank you, Rick. That's a wonderful introduction. Thank you. Let me see, make sure I can share my screen here. All right, can you see my actual screen?
Yep.
Okay, so I want to tell you a little bit about our work, primarily focused on utilizing non-canonical amino acids. And so I'll tell you this sort of theme that we've sort of generated in the lab. It sort of uses this tool called FEncAA. So this is fluorescence enhancement using non-canonical amino acids. And screen's working. Good. Okay, so sort of overall research themes in the lab, we work on large proteins that either have multiple domains or multiple proteins that function as large assemblies. And the work sort of focuses on proteins involved in DNA repair and recombination, like Rick mentioned. We work on proteins associated with nitrogen fixation and chlorophyll biosynthesis as part of our DOE-funded work. And there's a new push in the lab to study multi-domain RNA binding proteins that I'll touch upon today. So we are not GCE developers. We are actually, we love using it for many applications. And so sort of the bread and butter workhorse for us is sort of getting functionalized proteins, primarily that can be used to click chemistry and attach fluorophores to it. And then we can use it to interrogate domain dynamics. So I'll touch back to this point. So GCE has been amazing to us. We started working with GCE and Ryan and Rick, back in 2013, and it has allowed us to play around with a toolkit that was never available to us before, right? So most of our proteins are large; they contain multiple cysteines, and so interrogating what each domain at any given point does through unnatural amino acids was just eye-opening and amazing to us. So thank you for sort of helping us for all these years, and it's been just such a wonderful journey going forward. So I'm going to tell you about some of the work on RPA today, but I can tell you that we've made fluorescent versions of 52, 51 RNA binding proteins. We've made these proteins for other labs, and I think the last count that I have, I think we had 20 or 25 different proteins where we sort of engineered fluorophores into. So it's been versatile, it's been amazing, and really has set up almost everything we've been doing in the lab now. Okay, so I'm going to tell you a little bit about single-stranded DNA binding proteins. These are essential proteins that are required for DNA metabolism, and they are present anywhere from phages to mammals. We all have SSBs. And they come in a variety of shapes and sizes. So here's a monomer of a phage SSB protein. In the case of E. coli or in bacteria, they're either homodimers or homotetramers. And in higher eukaryotes, this protein called Replication Protein A, or RPA, takes over the function. And as you can see, it's composed of multiple domains that sort of function together. All right. So RPA is, as I said, essential. and it coordinates all the DNA metabolic processes, including all the repair processes, homologous recombination, nucleotide excision repair, base excision repair, mismatch repair, non-homologous end joining. It's essential for DNA replication, for fork reversal repair, telomere maintenance, cell cycle progression, meiosis, DNA damage response, and then, of course, genomic stability and associated cancers. So the holy grail question for us has been, how can a single protein play so many different roles? And how does this protein actually define specificity to these reactions? So the way you think about it is, you have a piece of double-stranded DNA and you open it up, and then you have single-stranded DNA. And that is the template onto which you are either replicating the DNA or repairing the DNA or recombining. And so when you open up the single-stranded DNA, RPA comes in and binds the DNA first, and it binds with very high affinity. And when you have long stretches of DNA, it forms these RPA filaments, which actually are triggers for cell cycle arrest. Okay, so if the single-stranded DNA is coated by RPA, then RPA dictates which of the next enzymes gets on the DNA. So that's where accessibility comes into place. Because RPA binds to DNA with very high affinity, how a lower affinity enzyme displaces or remodels RPA is an important question to address. So on top of that, we have issues with order and timing of how these proteins come in, when they come in, and where on the DNA in terms of polarity. Are they coming in, getting access to the end of the single-stranded DNA, or somewhere in the middle, or are they getting towards these junctions? And all of these properties are dictated by the intrinsic dynamics of RPA. And the reason I say dynamics is based on the sort of the overall structural organization of RPA. So there are 3 subunits, and it's always a constitutive primer. One is RPA 70. This is the large one. There is the middle one, RPA 32, and then the small subunit, RPA 14. But a better way to look at RPA is in terms of its DNA binding domains and protein interaction domains. And so these are OB folds that are present in RPA. So there are 6 of them: A, B, C, D, E, and F. And they can be classified into protein interaction domains and DNA binding domains. A, B, C, and D are DNA binding domains. PIT70N and PIT32C, also called F, and the winged helix domains are protein interaction domains. And then interactions between C, D, and E hold the trimer together. And these domains are actually connected by intrinsically disordered linkers, which makes it a lot more interesting because the protein becomes very floppy and can adopt a variety of configurations. These protein interaction domains are also pretty neat because each of them can bind to a select subset of proteins. So F can bind to many different proteins in replication and repair, and the winged helix domain binds to a unique subset of proteins. And there are proteins that also bind to multiple regions in RPA as well. So, how RPA is sitting on the DNA, how these incoming proteins work with RPA, and how they are accessing the DNA is pretty much what we are trying to answer. All right. So, if you were to consider those flexible linkers, you can actually do an MD simulation. So this is from Walter Chasen's lab, where they've essentially fixed one domain and allowed all the other domains to sort of float. And you can see the unique number of configurations that RPA can actually adopt. So it's a nightmare of a protein to sort of functionally interrogate. So we were sort of naively trying to address, what conformations are stable, which of these are more important. Are there specific configurations that are specific to certain functions? How are these configurations actually changed depending on the DNA substrates they're binding? So essentially context-dependent conformational formation is another question we want to sort of address. Okay, so here's a historical perspective and sort of why we got into RPA to begin with. So for the last sort of 2-3 decades, people have assigned different properties to these DNA binding domains. DNA binding domain A and DNA binding domain B are considered high affinity DNA binding domains, compared to the trimerization code, which was considered to be the low affinity binding domains. And these assumptions came into place because when people started studying RPA, since all the domains will bind to DNA, the only way you're going to interrogate them is by chopping up the domains and isolating the individual domains and characterizing the DNA binding activities. So when you do that, depending on the behavior of these domains, you can assign these affinities. And so in all the literature, if you read about RPA, they'll always put A and B first on the DNA because they are the high affinity domains, and the tri-C would sort of hang around or come in later. So So we were curious about, does this model apply in the context of the full-length protein? And the reason we asked that question is, if you take the crystal structure of RPA, A and B can only bind to 3 nucleotides each. The trimerization core actually binds to close to 15 or 16 nucleotides. So from that perspective, if you think about it, the trimerization core has more contacts on the DNA. So you'd actually expect that to be a lot more stable on the DNA compared to A and B. So this is where the challenge came in. So then can we actually study what these domains do, but by keeping the full-length protein intact? So this is sort of where we started off with. And so we turned to this methodology called PIFI, where you can put a fluorophore either on the protein or the DNA. And if you actually put the fluorophore on the DNA, if you have a protein that binds, the local environment of the fluorophore would change, and you can monitor the change in fluorescence. But the problem here is you're only going to get a readout from the end of the DNA or where the fluorophore is positioned on the DNA. And if you have multiple proteins, you have no idea which part of the protein or which of the proteins is influencing the fluorescence change, right? So it's got its limitations. Alternatively, you can actually put the fluorophore on the protein of interest. And so as long as you can position the fluorophore at the right place, then you can get a readout from the protein itself, and sort of shown over here, right? But this is really powerful because you can engineer one fluorophore on one protein, and no matter how many other proteins you add in there, you're getting the readout from the protein of interest. So this is what we wanted to do with RPA, where we want to go in and engineer fluorophores into unique positions onto the domains of RPA, and then get the readout as these domains are actually binding. The problem was, again, like I mentioned before, RPA has 18 cysteines, so there's no way you're going to go around trying to find a single cysteine to modulate it. So we turned to non-canonical amino acids. Again, I won't go into the details. You've been hearing about it everywhere. But the non-canonical amino acid we've been using quite a bit is 4-residue-phenylalanine. So you engineer the stop codon at a particular position on the protein where you want to engineer this unnatural amino acid, and we've done that here. And then shown here as an example with a protein called RAD51. So you express your plasmid of interest carrying the TAG along with the tRNA synthetase, you grow it, and then your unnatural amino acid gets incorporated, and then you can purify the protein and modify it or attach a fluorophore onto it. Okay. So long story short, it took us about 2.5 years to do this. We have been able to essentially engineer a fluorophore into DNA binding domain A at this particular position, or at position DBDT over here. And we've used click chemistry to attach a dye called NB543 or CY3 down the road to get the fluorescent protein, right? So there are two challenges, or at least two challenges that we had to overcome. One, we had to get site-specific labeling. Number two, the protein that carries the label should be almost as active as the wild type protein. So you have to not only find the position that doesn't affect function, you also need to have it accessible and be able to not put up structure. Finally, the protein has to give us a readout, a PIFI signal. So the position has to be close enough to the DNA or around the DNA such that when it binds, you see a change in the fluorescence. So it's a challenging sort of set of questions or targets, but we were able to do that. So there are some limitations to using this approach. So our protein yields certainly drop. We have about 60-70% loss in the overall protein. For RPA, you cannot use copper chemistry, so you have to use strain promoted click chemistry to do this. Our labeling efficiencies are still on the lower end, and it's still around 30 to 80% depending on the positions you're trying at. And we've had to extensively optimize buffers, temperatures, cell lines for each one of the proteins that we've sort of worked with. If you can't make the unnatural amino acid yourself, it actually tends up being quite expensive. So we've actually now been able to, we have very simple chemistries to make 4-residue-phenylalanine. So it's become a lot more accessible to us. Okay, so here are the experiments. So we've got two versions of RPA, one where the label is positioned on DNA binding domain A or DNA binding domain D. And so here was the really coolest experiment that I've seen in a really long time. No DNA. When you add DNA to the reaction, this is a stop flow experiment. You get the kinetics of DNA binding domain A engaging with the DNA. And when the label's on D, you actually get essentially the binding kinetics of D. So once you have active protein, once you got these probes, then you can go to town looking at the dynamics of the domains. All right, so I'll sort of walk you through about 5 or 6 slides of sort of summary slides of the data. So this one, for example, is again a stop flow experiment. We're looking at changes in either A or in D, and we're adding in increasing concentrations of a small piece of DNA. So the take-home message here is, of course, we can get the kinetics, we can get the on rates, we can get the off rates, you can model everything you want. But what was really cool was, A, you can actually see it binding and being remodeled depending on the concentration of RPA. D never got remodeled. So it feels like D wants to stay on the DNA all the time compared to DNA binding to my name. So this was the first evidence that A and B they can be high affinity, but they're definitely more dynamic than the trimerization core for sure. So here's another experiment where we sort of delete this trimerization core, and we're only looking at FAB. Again, the fluorophore is now on A. And so here again, you can see in the full-length protein is getting remodeled, but when you do not have the trimerization core, this FAB domain can actually occupy the DNA. So this is telling us that the protein can bind, but the trimerization core will eventually move in and displace AB from actually binding the DNA. So we're always now thinking that the AB is more dynamic. It's actually more off the DNA than on the DNA at any given point. Okay, so then having this tool allowed us to do single molecule experiments where we can actually watch one of these domains essentially go on and off, how many configurations and conformations it's actually accessing, and we do this using a single molecule TIFF microscope, where we've got a glass slide, you've got the DNA being attached, and you flow in your protein, and you're essentially watching the change in this fluorescence. And you can see your traces sort of look like this. So you can get it for A, you can get it for D. And these experiments were, the first time when we did this in collaboration with Maria Spies and her grad student, Colleen Colbert, up at Iowa, University of Iowa. So for both A and D, we could actually capture 4 distinct steps for each of the DBDs. But here is the power of this approach. So now you can actually add in RPA interacting proteins and see which of the domains are getting remodeled. So an example here is adding in a protein called RAD52. It is a recombination mediator that has to remodel RPA in order to get access to the DNA. And so when we do these experiments, what we really see is that there are 4 states for A. In the presence of RAD52, these 4 states do not change. So RAD52 binding does not change the dynamics of DBDD. However, when you look at DBDA, when you look at DBDD, these states actually change in the presence of RAD52, and this 4th state is completely eliminated. So this actually was the first evidence that when RAD52 comes in, it gets towards the 3' end of the DNA and selectively remodels one domain of RPA. So, I mean, if you think about the really coolness of this sort of setup, here's a protein that's got 6 domains, and we're literally getting a readout from one domain moving on and off the DNA. So to us, it was pretty phenomenal. We spent a lot of time trying to get fluorophores in each one of the domains. We were very successful right off the bat in getting DBDA and DBDD labeled. DBDC and B, we can get a lot of good labeling, but no matter what residue we touch, we lost protein activity. So you're still limited by what the protein gives you, and it needs a lot of trial and error for this to work. Okay, so that was one RPA molecule on DNA. And so now we can actually start looking at a filament where multiple RPA molecules are actually functioning. So the way we do these experiments is, now we've got a fluorophore positioned on DBDD on one half of RPA molecules and DBDA in the other half of molecules, and you can mix them together. And as they are binding and getting remodeled, you can actually watch Fret signal changes between the two. And so here we've done the experiment on a shorter piece of DNA. You can see, protein molecules actually binding. When you actually start adding more number of RPA molecules, then what happens? You actually see binding and you see displacement of A. So what we are thinking is going on is, as RPA molecules are assembling this filament, it is not ABCD, 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 but it's trimerization core, trimerization core, trimerization core, and AB is actually hanging out in solution. And we actually have now cryo-EM evidence as well showing that this is what happens. So, it's a pretty powerful approach to do that. Okay, so using the same approach, we've now looked at how these domain dynamics are modified by a chaperone protein. This guy is called RTT105. It actually binds to RPA, forms a 1:1 complex, brings it into the nucleus, right? So the idea being that when RPA is in solution, it can bind to 3 dozen proteins. How does the cell control binding between these proteins in the absence of DNA? So the answer is RTT105. It actually binds, and we ended up showing that RTT105 will hold all the domains together. And when you come in with DNA, each of the domains will be selectively released from RTT105, activating the protein. And we also actually showed, using the same Fred experiments that I just described, that if you challenge this complex with free RPA, it'll actually displace. So you can actually see a loss in the Fred signal. And that's a process called facilitated exchange, where RPA simply diffuses away. However, when you come in with RTT105 bound RPA, it is not able to challenge this filament. So this RTT105 holds complexes or staples RPA and sort of keeps it in an inactive state. And we were able to sort of uncover that using this experiment. So in addition to doing sort of simple ensemble experiments, we can actually do single molecule C-trap experiments now with the labeled RPA. We can actually watch multiple molecules of RPA on the DNA. And here we essentially showed that with RTT105, it cannot exchange. So, so there's a lot of tools that's been available for us to work with. So what I've showed you thus far was all the yeast RPA. We've actually now done the same thing for human RPA. And we uncovered this new phosphorylation by Aurora kinase at a single site in RPA. And we showed that this particular phosphorylated RPA can be more easily removed from the RPA filament. So again, this FRET assay is developing. It's got a lot more applications, and we're now starting to look at FRET between RPA and RPA interacting proteins on similar assays. Okay, then last 2 minutes, I'll just talk about some unpublished work again. So the ability to move the fluorophores around RPA now enhances our toolkit, because we can now start looking at fluorophores positioned on different places on the DNA and specific spots on the RPA. So here is an example where you have a 3' end where A is labeled, the A is labeled, and the other fluorophore is actually at this junction. And when you do this, you actually see the AB being kicked out because you do not see a change in the FRET signal. When you do the counter experiment where you actually have the label position over here at the junction, and D is labeled, now you see high, very stable FRET states, right? And when you do not have the trimerization core, this FAB, for example, just jumps around. So this idea that the A and the B are the high affinity domains, does not apply at all. The trimerization core is staying on, AB is hopping around. So we are trying different DNA substrates right now, trying to understand how RPA sort of functions over there. Okay, so here's a summary of everything we've done, for the last 10 years, and every single one of these projects has been facilitated by our ability to do GCE and incorporate non-canonical amino acids. So, so I'm happy to talk about with you any of these. Shoot me an email. I'm always happy to discuss the work. Okay, so this is now TDP2 run by Rick. That new phosphoserine version that he's developed. We've actually applied to this class of oncofetal RNA binding proteins called IMPs. Very similar to RPA, they have 6 RNA binding domains connected by disordered linkers, so they can sort of adopt different configurations. And that somehow drives RNA binding or RNA recognition specificity. Using GCE, we've been able to now fluorescently label selective domains in there, and we're starting to characterize it. And so we've also been able to get 100% labeled, phosphoserine labeled IMP1 and IMP3. So it works stunningly well, and we're excited to sort of explore this particular protein. Okay, so with that, I will just thank all the people that have done the work. A lot of this was done by my former grad student who's now at La Ronde, and then by Sahiti, who's now supported by an F99 from the NCI. And a lot of her publications to the GCE is really what enabled her to get that. And then, of course, funding for our lab and for the GCE team. Oregon. So saying that, we have lots of opportunities. We're looking for grad students, postdocs. So if any of you sort of want to come in and be part of our team, please reach out. Okay, I think I'll just stop there and open up for questions. So I think I'm on time.
Great. Thank you so much, Edwin. That was a really fantastic talk. So we'll, as you said, we'll open it up for questions. So if you have a question, either raise your hand in the Zooms or enter it into the chat. We can. Let's see. So Ryan's got a question.
Edwin, one thing that I probably should know by now, but I don't, is that with all of these sort of very significant conformational changes that are very sort of almost ordered conformational changes, I'm not sure what's driving those conformational changes. Are there ATPase cycles, or are there GTP cycles, post-translation modifications being driven on and off, right? So what's fueling this sort of ordered conformational change cycle system that you're looking at?
Yeah, no, so it's a great question. So I think they are dependent on the context, right? So normally, if you think about RPA, all these domains are sort of floating around is what you'd sort of assume. In the yeast system, the protein is regulated by RTD105, and it has to be released at the particular scenario by the incoming DNA. So depending on the context of the incoming DNA, a 3' overhang, a 5' overhang, a fork, or a gap, the configuration that's formed by RPA, so we call these configurations, right? So these configurations formed by RPA will be different and dictated by the DNA. And of course, we believe that in addition, they can be fine-tuned by intrinsic phosphorylations and other modifications for RPA, depending on the cell cycle context. In the case of the human system, we find something slightly different. We find interactions between these F domains and the wing helix domains all sort of binding together. So RPA is not really floating around, but it's actually more like in a ball to begin with. And then depending on the DNA and the PTM, you actually change. So interesting. I don't know if I have a slide here for the aurora kinase one with the human RPA. It's a single substitution in the middle of the protein that completely opens up these conformations, as if it's activating both protein-protein contacts and diffusion of RPA. So we've been fascinated by all these changes that we never even thought about RPA. And so I think we are slowly pegging around what these configurations are and how they relate to function.
Cool. John?
Yeah, that was really interesting. So you kind of answered this, but talking about DNA structure and how it influences all these interactions. So how did you go about picking your DNA sequence to start with, to just even get at this work? And then where are you headed? Because it's like almost infinite amount of combinations that you can do here.
Right. So the good thing about RPA is that it's a non-sequence binding protein, right? So we've sort of mostly used poly-DT so to keep away the secondary structure part of it. RPA can actually even unfold things like G quadruplex by simple diffusion, for example, right? So when we say context, we actually almost ignore the sequence. So if you're using like a 3' overhang, we'd use a double-stranded DNA that's base paired, and the single-stranded DNA will be poly-DT, right? So that's the way we think about it. So as far as we know, we can tell sequence does not play a role in how RPA binds. Now, the RNA binding protein is where we are now encountering this issue, but it's actually an exciting issue, right? Because with IMPs, there are 6 domains. They recognize up to 1,000 different RNA substrates. And there is no consensus that are, you can be like, oh, there are 3 or 4 nucleotides that may drive binding, but they're not the holy grail rules to hold them together, right? So they can be moved in. Multiple IMPs can actually bind to the same RNA depending on the concentration, right? So if you take, so what we decided is like, we'll keep RNA the same. We'll keep one RNA that we like, right? We'll either move around the sequence, mutate the sequence, and follow the domains. We can add in microRNAs to then regulate binding regions to different parts of the RNA. And then we're going to watch what these domains do. And does the affinity change? Does the configurations change? So all of that I think can be explored.
Yeah, thank you.